Hello, welcome everybody. It's my enormous pleasure today to host and chair this event. I'm really happy that so many of you have joined us today. I believe there's over 300 alumni and friends from all around the world joining today's webinar on Bringing Birmingham to You, Breakthroughs in Cancer Research. I'd really like first to make a big thanks to the donors. Many of you are our donors, and since we launched this campaign, we've already had over £100,000 donated to cancer research in Birmingham from 900 donors, and many of you are first-time donors. So thank you very much for your support, and I hope the discussion today will give you some insights onto where that funding is going. We can't do this research without your generous donations. In a moment, you're going to hear from two of our leading cancer researchers in Birmingham, Professor Andrew Beggs and Professor Colin Watts. You may have also been expecting Dr. Frank Masai to join us, but unfortunately, due to unforeseen circumstances, he's had to withdraw at the very last minute. But for those of you wanting to hear a bit about our childhood cancer research here in Birmingham, I will try to step in and talk to you on that topic. The running order today is each of our speakers are going to give you a little update on their areas of expertise, and then we're going to move into a panel discussion and then into a question and answer session with questions from yourselves. So I need to start with a little bit of what's called Zoom etiquette. So just to remind those of you who haven't had the message already on your screen, this webinar is being recorded. If you get the little message coming up saying that, could you just click on continue and then that message will disappear. While we're talking, as you get ideas for questions, please submit your questions. There's at the bottom of your screen, there's a little box saying Q&A. If you type your questions into there, we'll invite you to pose your questions to the panel. And if you don't want to do it through the Q&A, you're very welcome to do it through the chat, and we can respond online to the chat as well. All your cameras and mics are already disabled, but if you do want to ask a question, we'll reactivate your microphone and camera so you'll be able to pose your question. So without any further ado, I would like to introduce your panel. So first of all, I'd like to introduce Professor Andrew Beggs. He is Professor of Cancer Genetics and Surgery at the Institute of Cancer and Genomic Sciences at the University of Birmingham. He's got a long list of different responsibilities. He currently holds a Cancer Research UK and Royal College of Surgeons Advanced Clinical Clinician Scientist Awards. He's also a practicing clinician. He's a consultant, colorectal and general surgeon at the Queen Elizabeth Hospital here in Birmingham. And he specialises in laparoscopic rectal surgery, cancer surgery and advanced endoscopy. He's also the clinical theme lead for cancer genomic research theme within the Cancer Research UK Birmingham Centre, and he is our lead for translational biology and genomics research within the Institute of Cancer and Genomic Sciences. And finally, he's the Research Director for Cancer for the NHS South and Central Genomics Medicine Service Alliance. Hard to know how you fit all this in, Andrew. Could you just give us a short update on your research?
Thanks, Pam. Yeah, thanks very much for the introduction. It makes it sound like I've got about a million titles, but my main focus is on cancer. And we, thanks to the generosity of supporters up to this point as well, particularly Kern, Lee, Hancock and the Byrne family have been carrying out, and Cancer Research UK and the university have been carrying out a wide programme of research around various cancers. But the main focus of my research is bowel cancer. And one of the things we do is develop cancer models that are called organoids. Another word for them we call is avatars. So instead of treating a cancer very broadly, we take small samples of a patient's cancer and grow them into tumour organoids outside the body in our laboratory. And from that, we can give them drugs and treatments to understand why and how they're resistant to therapy and how we can improve the therapies we give to patients. As part of that further research, we're particularly interested in a type of bowel cancer called BRAF mutated colorectal cancer. And this is a small subset of bowel cancer. It's about 10% of patients with bowel cancer will have this, but it's very aggressive and it seems to affect young people more. And thanks to the generosity of supporters, we've started a research program here, which has funded a PhD student in my laboratory to carry out research to understand why these tumours are so resistant to treatment. Luckily, new treatments have come along for BRAF mutant colorectal cancer, but we're still not there. They don't work on everybody. And part of the research we're doing in my laboratory is to understand why these treatments don't work and see how we can improve them to improve survival from this disease. We also do other research more broadly around cancer. We're interested in other types of cancer such as esophageal cancer, so gullet cancer or stomach cancer, sarcomas, which are very large soft tissue tumors that affect adults and children, and tumors like thyroid, lung, breast, and other cancers that we all develop into models. Part of my other expertise is around genomic medicine, and what that means is we're trying to change the model of care such that instead of having very broad treatments, we're working with the NHS, can take small samples of your cancer and analyze them genetically to understand what makes them tick and choose the right chemotherapy agents for them. And that's being rolled out within the West Midlands and now across London, Oxford, and all over the UK. Finally, as well as that, we also participate in genomic medicine trials. So clinical trials for cancer drugs are moving more away from giving drug A for condition B, but to very precisely targeting certain types of cancers with the right drugs based on their genetic data. And we act as my lab as a national and international now centre of excellence for these types of trials, understanding which drugs we should give to patients and how we can maximise response for minimising toxicity. So you can see we've got a busy lab, we're quite broad, we do a lot of interesting things, but it's only really thanks to the generosity of supporters that we can continue to carry this kind of work out.
Thank you, Andrew. There's a lot we can dive into shortly on that, but I'd like now to just introduce Colin Watts. So Colin is a professor of neurosurgery and he's also a practicing clinician. He's an honorary consultant neurosurgeon at the Queen Elizabeth Hospital. And Colin is leading a newly established brain cancer program at the University of Birmingham, where his research is aiming to improve the treatment and survival for patients with glioma. And I think I shall leave Colin to really give us the details of that research. He's got a particular interest in intrinsic gliomas and cerebral metastases. And Colin, I will let you expand on that research. Would you like to give us a bit of an update on what you're up to now? And you're on mute at the moment.
Thank you, Pam. And I would echo our thanks to all the donors, including the first-time donors. It's great that you're making the initiative. The brain is the only thing in the universe that presumes to study itself, at least the only thing that we're aware of. And one of the areas that it's particularly bad at studying is how cancers grow in the brain and where they come from. We are developing a program and it has been enormously supported and helped by some of the generosity from our philanthropic donors around three areas. One area is understanding how these cancers grow. Another area is understanding how we can improve our treatments for these cancers. And the third area is understanding how we can support and help patients in their journey to learn how to cope with these cancers and to deal with all the stresses that come with the treatment. How these cancers grow is an interesting one, and we don't really understand it very well, I'll be honest with you. By the time brain cancer presents itself clinically, either through a seizure or through changes in behavior or through apparently having a stroke-like weakness or something like that, it's quite advanced. And that is one of the challenges. So if you imagine the Earth when the dinosaurs roamed the planet, and our treatments are like the meteorite that hits the Earth, wipes out the dinosaurs. And all that does is clear a pathway for these previously unseen animals, the mammals scurrying around in the undergrowth, these stem cells, these cancer promoting cells. It gives them space to take over. And they grow and they spread, and then they dominate and drive the tumor. And some of our work is related to understanding how that driver domination develops and how these different cell populations, like animals on Earth, fight for resources and compete to outdo each other and grow. We're doing that at the moment primarily from the perspective of tumors that arise de novo from the brain. But increasingly, we're interested in tumors that spread to the brain from elsewhere. And our top three are lung cancer, breast cancer, and skin cancer. They're the commonest ones. In addition to that, we're also running something called the Tessa Jowl Brain Matrix. It's named after Baroness Tessa Jowl, who herself died of a primary brain tumor called a glioblastoma. These are the commonest and most aggressive brain tumors. And she gave a fantastic rousing speech in the House of Lords, where she asked for effort to promote hope so that every patient is looked after while the scientists work hard to try and develop new treatments. Within the brain matrix, we are looking to develop new treatments and get them to patients. We're also looking to capture all the data, the imaging data, the clinical data, and the biological data, including the genetics data that you've heard about already from Professor Beggs. And that trial has now started, the platform trial has started, beginning to develop that infrastructure to get genetic and imaging information back to help patients treat patients better, sorry, to help clinicians treat patients better. And lastly, but by no means least, we're working on research to try and help the patient in the here and now, to help them cope with the journey, to understand what things matter to patients, whether it's managing seizures, coping with the side effects of drugs, or coming to terms with the inevitable end, which is the recognition that we're all mortal. So those are the three areas that we're interested in. It covers a holistic approach, and we're building a fantastic team. It's worth mentioning that we're working with Andrew on, utilizing his organoid skills and stuff like that to develop what we call patient avatars, so that we can quickly test in the lab the drugs that we propose to treat the patients with. And actually, in an ideal world, get the cells from the patient and test them with drugs that we're going to try in that same patient, having a proper patient avatar. That's something that we're working on in the future. So that's us. Thank you.
Genuinely innovative and exciting work, Colin. Now, we were going to hear from Frank about his work on childhood cancers, and perhaps if I could just speak to that a little bit. So I am a professor of paediatric oncology at the university, and I work as a paediatric oncologist at the Birmingham Children's Hospital. In my hats at the university, I'm director of the Cancer Research UK Clinical Trials Unit. And our clinical trials unit is the national trials unit for childhood cancer trials. And the challenge we face in paediatric oncology is that childhood cancer is rare. Now, in some ways, that's not a challenge. It's good that not many children develop cancer. But the number of different cancers that children get is very diverse, and they are not mini versions of adult cancers. They are unique diseases that happen in young children with a very different reason for developing. And the treatments have basically been, if you like, hand-me-downs from the adult world up to now. We use really toxic chemotherapy drugs, surgery, radiotherapy in children that were really originally developed in the adult world. And so the challenge we have is that the toxicity that children experience because of this treatment not only impacts on the time when they're on treatment, and therefore it impacts on their development and their school lives, but also a third of children that are treated for childhood cancer go on to have long-term side effects that have an impact on the way they live their lives in the future. So what we're trying to do is develop new treatments that are not only effective, but have less side effects in the future. And the way to do that is to have a better understanding of the biology of childhood cancers so that treatments can be more targeted. But when you try to translate the work that's being done at the bench into clinical trials in children, we have to recognize the numbers of patients are small. So to get good scientific evidence, we need to do collaborative research. So nearly all our research is international. 98% of our clinical trials that we run for children involve other countries. And also we have to design them in a very innovative way. And we're very lucky in our trials unit to have incredibly innovative statisticians that design trials that answer research questions with the smallest number of patients possible. So that we can get to the answer for the improved treatment for children at a much earlier stage in a drug development. And we're now working on some really quite innovative models, partnering with industry. Industry haven't been interested in children with cancer because the numbers are too small for them to be economically interesting to large corporate organizations. But we're finding innovative ways to work with them so that we can deliver the trials in partnership with industry, such that the research questions can be answered for the benefit of patients, and the data can take those drugs into licensing into the NHS at a much earlier stage. So that's just a little bit of a snapshot of what we're doing in childhood cancer. So I'd like to open up some questions now to the panel, and I'm sure they're going to turn them back on me. But just starting, Andrew, what do you think in your area are going to be the big breakthroughs for cancer in the next 5 years?
Thanks, Professor Kern. So I think probably targeted therapy. So chemotherapy, there may well be people on this webinar who have had chemotherapy themselves or know somebody who has, and it's a pretty brutal drug. Colin's analogy of an asteroid hitting them is very true. It's a massive whack. We're getting much better now understanding and tailoring chemotherapy to the right patient at the right time and the right type of drug, so they can have a better response and less toxicity. And we're getting better understanding why patients get the toxicity as well. And that's happening in the here and now. The NHS is routinely adapting these things, but they can only do it through research, from us understanding why these patients are sensitive to these drugs, doing the trials to carry out and prove that they work effectively, and then also back with the scientists understanding why they don't work in some people, these revolutionary drugs. One of the examples is immunotherapy. Immunotherapy is a drug that unmasks cancer to the immune system and allows it to be seen and allow it to be fine. Actually, we now know what patients we should give immunotherapy to. It's an expensive drug and it doesn't work in everybody, but in people it does work. It can have a dramatic effect, effectively wiping out the cancer. And so one of the aspects of scientific research that we do is working with drug companies to understand why this is the case. And then working with our colleagues in clinical trials units, we take it forward into patients.
Thank you, Andrew. And I'm pretty sure there's very similar parallels in brain tumours, Colin. What do you think the biggest breakthroughs are going to be in the next 5 years for brain tumour research?
That's an interesting one. I think there are several things. But overarching one is data, how it is handled, how it is collected, how it is manipulated. And what I mean by that is bringing together, for example, the imaging that we do, the clinical information that we have, the genetic information that we have, and the understanding and the data relating to how those genetic changes translate into how cells actually behave and how these cancers actually grow. I think there's an interesting element, which is how the cancers interact with their environment. So, for example, in the last 2 years, it's been discovered that brain cancer is electrically active. It has synapses with the brain. It responds to the electrical activity in the brain. But we also know that there are things like how these tumors feed, how they get their fuel to grow, how they overcome the immune system, which is supposed to scan the body for all these cancers and things. So I think that's another a really important area. And one of the challenges that we have with brain is that it's very difficult compared, for example, to the liquid blood cancers or some of the easier to get to cancers like breast cancer. It's very difficult to get tissue. And I think building the infrastructure to link the people who can get the tissue in the clinic with the people who can do the breakthrough mechanistic research in the labs, building that infrastructure is also going to be critical. And it's worth pointing out, as you know, Pam, that is one of our priority areas in Birmingham. It's something that we're really developing. We're at the forefront of doing that. And that's been recognized, for example, with our recognition of Centre of Excellence for brain tumours and stuff like that. It's all about bringing the clinic and the lab together. So I think that infrastructure is also going to be critical.
You raise an incredibly important point there, Colin. I think, we often talk about the new scientific breakthrough in terms of the new omics, the new gene, the new therapy. But that, if you like, grunt work, that getting the sample out of the patient in a way that is still viable and as close to what it was like in the patient into the lab and analyzing it is a huge hurdle. And I think, as you said, the infrastructure that you're building is essential to allow the research to happen. I'm going to go a little bit off piece now and just go into the chat, because our very own David Adams, head of the Medical and Dental College here in Birmingham, has generously shared his story about his son, who was diagnosed with a WHO grade 2 astrocytoma of the brainstem. And with you'll see in the chat the mutations, the IDH mutant, BRAF wild type, ATRX mutant, MGM methylated. I'm not going to ask you, specifically, Colin, but having that snapshot of a tumour's genomic fingerprint, if you like, how is that going to help us get new treatments for young men like David's son?
So understanding that there are those genetic changes is the first step. The next step is understanding how those genetic changes result in how the cells behave. So those genetic changes have to be translated into a language that builds the blocks to drive the cell growth, and the blocks are proteins, basically. So understanding what those profiles are allows us to begin to think about targeting mechanisms. And it's likely that as a result of that understanding, we'll be able to develop those drugs that will target it. I actually think we talk a lot about targeted medicine, but that is a really challenging thing, because I do not think a single drug is going to be a magic bullet. I think what we're going to have to do is have combinations of drugs that are targeting different elements. So using that example, something that would target IDH, something else that would target BRAF signaling. But we're going to need to be canny about how we deliver these combinations, because it's not going to be give everything all at once. It's going to be learning how to deliver one, then the other, and small doses of two together and so on, because toxicity can limit that. And if we're going to do that, then one of the key things, the critical things that we need to do, is to be able to identify when treatments fail as quickly as possible, so we can turn patients into different drugs, different treatments.
Yeah. And I think the critical question is, Ros, Dave's wife, that's putting the questions in, is, what are we doing in Birmingham to advance that?
So in Birmingham, we're looking at how the environment, the fuel, the feeding, the growth, the metabolism, which is intimately related to the IDH mutation. We know that the absence of the IDH mutation is associated with very aggressive growth patterns, and the presence of the IDH mutation is associated with less aggressive growth patterns, but we don't understand really why. And there are other downstream targets, as well as drugs targeting the IDH mutation, that are in early phase development of clinical trials. And again, that's something that in Birmingham, we're looking to bring into the Brain Matrix program. At the same time, there are also techniques that we're using to identify where these, in the low-grade tumors, where these nests of cells that are going to change and drive growth of that tumor in the future, where they are. And we're developing techniques in the lab to look at that by utilizing some of the fluorescence technologies that we use in the clinic, in the operating room.
Okay, thank you for that, Colin. And I can turn back to Andrew, because we've been through a very difficult year and a half with the COVID-19 pandemic globally, but it's certainly had an impact both in the clinic and on research. Could you just highlight for people listening in as to what that impact has been on your research?
Thanks. So there's been a couple of impacts of COVID-19 on research. I think probably we're all a bit sick of COVID-19, but I think COVID-19 has affected people both personally and professionally. And from my point of view, it's affected me and that a number of my family have had, including my wife. And so it affects researchers as well as, amazingly, you just don't realize how many people have had the disease. But in terms of professionally, one of the issues with COVID-19 is that we've had to, for safety reasons, reduce the amount of research we're able to do because of social distancing requirements. We don't want people to get COVID. We don't want people to get past the infection in through the university. So it slowed things down a lot. And from the point of view of research as well, people are struggling to make ends meet because of losing their jobs. They are, less money's been coming, the economy's taking a hit. And as a consequence, people will donate less to charity, understandably, because when times are tight, you pull your belt in. And that's led to a decrease in funding. A lot of our major research funders that we rely on have tightened their research grant programs, sometimes up to 50%. And certainly on a national level, my own research grants have been cut from between 15 to 50%. One of my grants in particular on sarcoma, an international grant with collaborators in Italy and Spain, was cut by over 50%. And it makes it very difficult to carry out the research you want to do. But you understand why it's happening. And this is why the role of alumni fundraising is so vital, because we can talk to you directly, you can understand where we're coming from, what research we're doing, and that is thanks to generous support like that we've carried it out. I mean, COVID has had a an upside in that my laboratory, because of our expertise in molecular genetics, has been deployed to helping the government with COVID testing. And so when there were struggles at the beginning of the pandemic in getting people access to testing, we repurposed the equipment to allow it to happen. And that was able to help the local NHS quite considerably. And we've helped develop the NHS Signals and Tests as well. But I mean, COVID has had a massive impact. Cancer research is suffering because of it. So even something as simple as international travel, a lot of cancer research happens based on international travel. People speak to each other. We work with collaborators. We can't do any of that now. And Zoom, it does allow it to a bit, but there's nothing quite beats being able to get together in a room with some people and throw some ideas together about how to help people with cancer. So it's had a lot of effects. My main hope is that it looks like the economy is going to rebound quite profoundly. At that stage, I'm hoping that people will be able to think about contributing to charitable research in cancer in the future.
Thank you, Andrew. I mean, actually, it's the unexpected consequences of COVID, isn't it? You think you can engage by Zoom, which we've all done incredibly well and probably saved the planet from not traveling so much. But on the other hand, those conversations that with your international collaborators that spark new ideas are really exciting, and we have lost a fair bit of that. I mean, Colin, you're a neurosurgeon, and the impact on surgery has been very much in the news. But how has COVID-19 impacted you in both clinical practice and in research?
So one of the things from a clinical perspective that COVID has done is it has reduced help-seeking behavior by people, by patients, which means that patients are presenting at later stages of the disease, which means that many are now inoperable. And so we've been struggling with that, both in terms of trying to help patients, but also, of course, on the psychological impact, both of patients and for staff in these terrible circumstances. In terms of the research, then, of course, the labs have been shut down. Operating means has been reduced, so we're not getting the tissue into the labs in the way that we were and that we will do when we go back to something more normal. I think the other thing that's worth bearing in mind, Pam, is that one of the things we've learned that's a positive from COVID-19 and the way that the research has been done to develop the viruses is that if you embed research in NHS clinical practice, it can accelerate discovery and accelerate validation and develop drugs and treatments much quicker. And I think that's a lesson that we're going to take away and try and apply and work out how we can do that. Because I think embedding research in a routine NHS practice is really important. And COVID's shown it can have incredible value. And of course, Brain Matrix was ahead of the curve in the sense that was intrinsic to our strategy long before COVID happened. But I'm hoping that maybe as a result of lessons learned, it will actually help us leapfrog forward faster than we might otherwise have done.
No, so that's a very important point. I mean, in clinical trials, we were, from some respects, our trials unit in the university coped with COVID reasonably well in that we pivoted all to home working very quickly. But what we couldn't do is help the hospitals who couldn't bring the patients in who were going on to clinical trials into the NHS to recruit them because of all the restrictions that had to happen and the pressure of work. So we definitely saw recruitment into clinical trials dip, and also being able to open new trials is really challenging, and we haven't fully recovered to that yet. But some of the imaginative things that were put in place for how we do online consent, how we do online clinics, actually will help in the future. And one of the things that really is frustrating in clinical trials is speed. And the COVID-19 pandemic showed a way of speeding up clinical trial set up from a regulatory point of view. So that, as you say, there may be lessons learned from that. So I'm going to give some final questions to both of you, and then I'm really going to go to the Q&A because some great questions coming in from the audience. So just very briefly, just can you highlight the impact of the work that you're doing at the moment? Just a couple of points on what impact specifically the work you're doing is having.
I think the work specifically we're doing around BRAF mutant colorectal cancer is having an impact because we're getting to the stage now where we can better understand who will be sensitive and resistant to treatment, and also improving access to therapies for patients who have it. We're doing a similar sort of body of work in a different tumor type called cholangiocarcinoma, where we're looking at a slightly different drug, which will help us understand how we can better treat those patients. And I think just generally making tumor models like Colin and I both use and genomic medicine mainstream is probably the biggest impact of our work so far.
Colin.
So I'd echo that on the basic translational side and developing the patient avatar program. Our holistic approach to combining what matters to patients, to what goes on in the lab, to what happens in the operating room and the clinic, has ensured that we've achieved recognition nationally. We've been awarded a Centre of Excellence Award for brain tumours from the Tessa Jal Brain Cancer Mission. So that's excellent. We've also got a trial that is in the early stages of setup, where we're looking at cannabinoids and how they might help patients with brain tumours. What I find fascinating about that is the delivery mechanism, which is through the nose. So we'll have to see how that goes. But a variety of things that we're having as a result of what we're doing that's been supported, not least through our philanthropic donors. So thanks to everybody.
Fantastic. Just from the trials unit point of view, I just say a big thank you to Catherine. Delighted that you dialed into the webinar today. She highlighted the issue of long-term side effects from a son who was treated for rhabdomyosarcoma. We've just launched a trial, a global trial called FAR-RMS, which is for frontline and relapsed patients with rhabdomyosarcoma, to try and improve the treatments at a number of different levels. One is looking at the impact of radiotherapy and the timing of radiotherapy, which is not, and the dosing of radiotherapy, which is not optimized yet. And I think this will be a really important question to answer. But it's a complex, what's called a complex innovative trial, in that we've got lots of different questions within it. So we've got chemotherapy questions, radiotherapy questions, but we've also got questions for new drugs, bringing in new drugs into the trial that would otherwise not be investigated. And we're bringing a new drug in called regorafenib for the most difficult to treat rhabdomyosarcomas, to see if we can improve outcomes. So it's a really exciting launch in that disease area. And another trial that we've just completed, which is in a bone tumor called Ewing sarcoma, and is again talking to this long-term toxicity. It was a very straightforward question. We asked which was the best chemotherapy regime of standard chemotherapy drugs that we have, comparing the American standard with what was usually used in Europe, and asking the question is, can we work out which has got the least long-term side effects, short and long-term side effects, without impacting survival, because we were getting good results from the survival perspective. And in fact, we managed to determine a new standard of therapy for Ewing sarcoma at the beginning of treatment, which was actually turned out to be the American standard. But that is now being implemented as clinical practice globally, and it means that we can build all new trials on a new standard of practice that has less side effects than we had before. So I'm just going to bear with me when I just look at all the questions that are coming in, and I would like to start off with Ken Wigfield, who's actually posed a couple of questions. Ken, I think your microphone is being activated. Would you like to pose your question? I think we just need you to unmute, Ken.
There we go. Can you hear me now?
Can hear you.
Okay. Right. We tend to hear about cancers, and you've spoken about them, and they seem to be a rather chaotic, eclectic mix of these cancers. Yet, as a member of the public, you seem to hear cancer as a single entity. Is there any real relationship between these cancers? I tend to use taxonomy in some of the work I do. And so you look at the structure of things, the structure of living items in a taxonomy, in this hierarchy relationship. I wondered if such a relationship existed within the cancer regime, as opposed to being just a whole set of different diseases.
Andrew, would you like to try and take that, and then I'll hand over to Colin?
I like a good scientific question. Thank you, Mr. Wigfield. So cancer, I suppose the broadest way to define cancer would be abnormal growth of cells. That would be the catch-all. But every cancer is different at the tissue level because you require a different genetic change to start it off. So the change you see in a bowel cancer when it starts off is different to say in a brain cancer or a lung cancer. But once it gets that first initial sets of changes, what happens with cancer, which is why it's so resistant to treatment, is, if you want the technical term, branch Darwinian evolution. So in other words, the tumor evolves to survive in the body and resist treatment. And every time you give it a treatment, say chemotherapy, radiotherapy, it resists to evolve that. So it evolves to resist that treatment. And so in terms of taxonomic classification, if you take, say, a metastatic cancer, one that's spread, and compare it back to the primary tumor it came from, you can see they're related, but they're related down many branches down a taxonomic tree, which has taken many subclones that have died off and many clones that have asserted dominance. So cancer, unfortunately for us as humans, is the ultimate example of evolution. And that's why it's so difficult to treat.
Right. Thank you.
Thank you, Andrew. Colin, do you want to add to that?
Yeah, so I think it's interesting, Mr. Wakefield. The way we think about cancer has changed significantly in recent years by applying Darwinian evolutionary principles, as Andrew's just eloquently articulated. And it's interesting your comments about taxonomy, because obviously the early biologists, before we knew about genetics, were busy classifying things and developing taxonomic measures. The challenge that we have is understanding which of the populations. So think of cancer, each individual type of cancer, as Andrew said, is different from others, a bit like different species, different countries, different continents on the planet, for example. But within each continent, there are similar niches. Right. And within those niches, you've got different types of animal occupying the same niche. So in one continent, you might have kangaroos. In another continent, you might have bears, for example, just as an example. Yeah. But our understanding of the behavior is influenced by the interaction between how those species grow, how they feed, how they defend themselves, and the environment in which they're growing, which is why, of course, you have extinctions. And when you have extinctions, then something else takes over, because that niche is simply cleaned out and repurposed, if you like. So that is increasingly how we're thinking about cancer. And how does that help us think about treatment is an interesting challenge, because as you target one population and get rid of them or reduce them, then another population grows and dominates. If you use agriculture as an example, one of the ways that they've got rid of weeds in crops without damaging the crops is by using combinations of weed killers rather than single doses of very concentrated toxic weed killers. And by doing that, they've kept the toxicities down and killed the weeds. We need to understand how we can do that kind of thing in cancer.
Yeah, lovely. Thank you.
Thank you. I hope that answered your question, Ken. So I'm going to now just quick answer to a question that's come in, just asking about the webinar's availability in future, just to let you all know that it will be published in YouTube next week for those, if you've got colleagues or friends that missed the webinar and want to listen to it. I'd like to bring in Rabia Kawaja, who's asking a question about big data. Rabia, I hope your microphone is going to be activated now. If you unmute, you'll be able to pose your question. Just click on the unmute so that Rabia can pose the question. Rabia, could you just click on unmute? It'll be at the bottom of your screen. You should be able to click on it. Rabia, while you're finding that unmute, maybe we'll move on to another question, then we'll come back to you. Maybe we could bring in Paul Isaacs, who's got a question around Brexit.
Hi, it was prompted by some of the comments made earlier about your funding. I was just wondering how Brexit was affecting your funding, both in terms of government funding and perhaps some of the France, Erasmus types, and that you've noticed any impact yet?
I'll maybe start with that. Yes, I think the short answer is yes. There's two impacts that Brexit has had on our clinical trials research. One is that because we're now a third country, it's impacted on the regulatory framework for conducting clinical trials, which meant that we've had to put in, if I could call it fixes, ways to allow us to continue to lead clinical trials from the UK within Europe, and that's added some, additional expense to and time to deal with those extra regulatory hurdles. And we've previously had really excellent funding from the EU Horizon programs, which has meant that we've been able to fund international clinical trials, both in the UK and internationally. And whilst we have been told by the government that we will have access to Horizon funding in the future, I think we're still waiting to see how that's going to pan out. But in Europe, they've identified cancer as one of their major initiatives. The European Beating Cancer Plan means that there will be a lot of funding available for cancer research. So I hope that we will be able to leverage that funding. I'm going to hand over to Colin. Have you had any impact from Brexit from your side?
Not really, to be fair, at this stage. We have funding that's around education and training the surgeons of the future, developing new training techniques and stuff, which there are 2 UK centres as part of a massive European consortium, and we're the lead for the brain cancer work. That hasn't been impacted yet. It's been delayed as a result of COVID, but the funding is still there and it's not been treated. I think the thing is with the paediatric community are much more involved in the international organisation and international facing work because of the small numbers in any individual centre, and therefore likely to be impacted. We are watching with interest to see what happens, and from my own perspective, huge gratitude to the trials team for jigging the regulations, which I think at one point were changing on a weekly basis. So that's as far as we've got so far.
I think, Andrew, you've seen an impact from the PhD studentships, haven't you?
Well, a couple of things from Brexit. One is yeah, students. We just don't get the EU students and postdoctoral scientists anymore because the visa. I'll be really blunt. The visa environment is too hostile for them to want to come now, compared to where it was before, and so they're less likely to come here. They prefer to go to the States and Australia and other places rather than come to the UK, which is upsetting. The other problem we've had is around cost of raw materials, reagents that we do to do scientific research. One particular thing we used to go grow the organ when we left the EU went up by 50%, which was a direct result of Brexit, which was quite depressing. So we've had to move to an alternate supplier, which in the end is fine, but it just means having to change quite a lot of workflows. And the other problem with Brexit has delayed common stuff coming across. We used to, as part of a free market, we used to be able to get reagents, equipment from Europe very easily. But unfortunately now it gets delayed, it gets stored in customs, or in a couple of instances has been sent back because, for instance, of import duty problems. So, I mean, these are all issues in the short term, and hopefully in the long term it will get better. But in the short term, it has affected us quite a lot. From the research point of view, we have had quite a bit of funding from the EU as well as part of the Horizon program. But going forwards, I believe we're going to be a part of it in the UK. So, we're relatively unaffected from that point of view. I think time will only tell how things will go and whether the scientists will return. But unfortunately, because of Brexit, the UK is not seen as an attractive place to work anymore for your PhD and postdoctoral scientists that we would have got before.
Thanks, Andrew. Unfortunately, Rabia's we've got some technical problems unmuting Rabia, so that I'm going to pose her question on her behalf. The question is really about big data. How is data science and big data playing a part in cancer diagnosis? And I think that's quite close to your heart, Andrew. Do you want to start with that?
Yeah, thank you. So, Rabia, I'm an Alan Turing Fellow. So you may have heard of may not have heard of the Alan Turing Institute. It's the UK's National Data Science Institute. And my area of expertise is around cancer genetics and integrating it back with patient data. And big data plays a massive, no pun intended, plays a massive role at the moment in diagnosis and treatment going forwards. So the UK has just completed the 100,000 genome project. So I was part of that. And that it looked at a lot of patients, about half the patients had rare disease, about 50,000 of the patients had cancer. And if you want big data, I just looked it up on the system now while I saw your question pop up. The 100,000 genome data, they've generated 55,000 terabytes worth of sequencing data over 4 years, which is 55,000 hard drives, and that's an enormously powerful data set because it's linked back to clinical outcomes, it's linked back to how patients respond to treatment, and we can already make new discoveries based on those data sets. But going forwards, a lot of hospitals are moving towards, in a different direction, moving towards electronic patient records. And that allows us to link the data back together, anonymously, of course, because there's been a lot in the press about it recently, but anonymously, so we can understand how particular treatments work for people and how their genetic information can be used to understand how treatments can be improved for them. So that is an enormously powerful, and actually that's something the UK does really well. We're far beyond any other country in the world, including the US and China, on this type of technology. And patients in the NHS on the ground are getting benefits from this every day.
Thank you, Andrew. Just mindful of the time, so we don't completely run out. There's just one other question that was raised about research going on to support for treatment for patient support after treatment ends. This person points out from a personal view that it's been harder than the actual treatment, and I think that's a comment that would resonate with a lot of people that have been through cancer treatment. And the long-term side effects research is something that we do need to focus on. In the University of Birmingham, we've got the Centre for Patient-Reported Outcomes Research, and this is a way that we can just start to better understand from a patient perspective, with information coming directly from patients, as to how the effects of chemotherapy or other aspects of treatment, whether it be radiotherapy or the surgical impact, is impacting on their lives, and allowing that real-time data to be fed into the way we develop trials and the way we develop treatments in the future. I'm going to wind up by just asking my panel for one quick, literally one sentence each, as to how philanthropy is helping you, and then I will close the meeting. So Andrew first.
It allows us to do things that wouldn't otherwise be possible in the current funding landscape. So thank you.
Colin.
It allows us to take risks and do things that are at an early stage and are challenging to get funded through normal routes.
Thank you, Colin. And I think from our perspective, I think the taking risks is definitely the one. If we can accelerate clinical research by being able to do the pilot studies and the development work before we can secure international or national funding, it allows us to move research forward much more rapidly than we can if we have to keep waiting for the long-term funding rounds. So that basic investment at the beginning is incredibly important, and we're very grateful to that from our philanthropic donors. So it just remains for me really to thank both the panel members, Professor Andrew Beggs and Professor Colin Watts, I think, for a really engaging discussion today. I want to thank everybody for attending Bringing Birmingham to You event on the breakthroughs in cancer research. I'd like to thank all of you who submitted questions. I hope we got through most of them. My apologies if we haven't answered your question at all or as thoroughly as you would like. If you're interested in finding more out about our cancer research and how to support our cancer research in Birmingham, a link to our website, I believe, is being put in the chat. Yes, there we go. It's great, and it happens just as you say it. We're going to continue with this series online. So if you're keen to hear more about areas of research, please look at the online offers that are coming through. And if you've got ideas, things that you'd like to hear about more, please get in touch with my colleagues at the alumni office, and we can arrange specific webinars on topics that you're interested in. I also want to draw your attention, when you go to the website, the University of Birmingham has recently launched a new program called the Birmingham Health Innovation Campus. And this has got within it something called the Precision Technologies Health Accelerator. And much of the stuff that we've talked about, which is a really fundamental research within the university, we're finding an innovative way to now link this with industry partners, biotechs, and small companies to really start to bring genuine innovation that can improve patient outcomes to the heart of Birmingham and the West Midlands. So I'd like, if I could draw your attention to that website on the which you can link to through the University of Birmingham website as well. Please send your feedback on this event. There's going to be a short feedback survey, which I believe will come into the chat as well any second now, and a QR code, thank you, and a QR code will be displayed on the screen. For those that submit feedback, you'll be entered into a prize draw to win a Z-Clay print from Brumhouse. And for those of you who have not seen these, it's worth Googling them. They're really interesting prints. And it's a fantastic one of the University of Birmingham campus. So thank you all again for your time, for your attention, and I hope you enjoy the rest of your day. Thank you.